So hello, everybody. Welcome to our webinar, Precisionary Instruments. And today we are honored to host Dr. Francesco Travascio, who is an associate professor in the Department of Mechanical and Aerospace Engineering down at the University of Miami, where it's nice and warm. So Dr. Francesco Travascio it directs also the Musculoskeletal Biomechanics Laboratory at the University of Miami. In addition, he also serves as Associate Director of the Max Biedermann Institute for Biomechanics at Mount Sinai Medical Center in Miami Beach. So in general, the meniscus is a fibrocartilaginous tissue in the knee that plays an essential role in load distribution, congruency, and joint stability. As you can imagine, all of us have been up and down on our knees. And accordingly, the presence of having a healthy and functioning meniscus is necessary for proper joint biomechanics and everyday enjoyment. Therefore, sometimes you might have or know somebody who have experienced a traumatic failure like a meniscal tear, which is the most frequent type of injury to the meniscus, particularly in active younger adults. The repair of tears using surgical approaches decreases the development of osteoarthritis and subsequent need for joint replacement, but its rate of success is severely limited. So the ultimate goal of Dr. Travascio's research is to develop a long-lasting treatment for meniscus tears based on regeneration of the tissue and preservation of its function. So in this talk, he will present his research advances in understanding the biomechanical behavior of the meniscus and its transport properties in relation to its unique compositional and structural features. So with that, Dr. Travascio, I would love to hand it over to you. And thank you so much for being with us today.
So good morning, and thank you very much for having me here today. And thank you for the generous introduction. So as you just said, I would like to talk about some research work that we have been conducted at the University of Miami. In general, we are working on a better understanding of the pathophysiology of the meniscus in the knee and on strategies for preventing the degeneration of this tissue. We are also interested in developing new biomaterials that can be used for a partial or total replacement of this tissue once it becomes irreparably damaged. But in today's talk, I would like to focus first on the most recent findings on meniscus biomechanics and biotransport in relation to the structure of the tissue. And we believe that this knowledge is crucial in order to develop successful therapeutic approaches to cure the degeneration or the development of surgical approaches for the repair of this tissue. So first of all, I'd like to provide some background information to those who may not be very familiar with the knee meniscus. This tissue has a semilunar shape and is composed of fibrocartilage, and we will see in the next slides what I mean with fibrocartilage. Its primary function is to absorb and redistribute the mechanical load on the knee that develops during any activity we perform during any exercise. The other important function is to maintain the congruency of the joint and its lubrication during motion. Now, the meniscus lives in a very aggressive mechanical environment. It is supposed to bear from 45 to 75% of the total load of the knee, which is equivalent to 2.7 up to 4.9 times the weight of the entire body. Think about in activities like jumping or landing, and these can be very aggressive mechanical loads we are subjecting the meniscus to. Now, meniscal injuries are among the most prevalent traumatic injuries that you can find in the musculoskeletal system. They generally present as tears along the longitude, the radius, or the circumference of the tissue. However, the longitudinal tears are considered the most common one. It has been hypothesized that the formation of the meniscal tears is associated to excessive mechanical load on the knee, as well as the structure and the peculiar structure and compositional features of the tissue. Now, the current approaches for the treatment of the tears include repairing or suturing. This is done when the tear is present in the outmost region of the tissue, which is vascularized and is also called the red zone. and I will discuss the meniscal vascularization specifically in the next slides. In any case, the failure rate of the tears is more than 20% at 5 years post-operative. And the alternative approach would be the partial or total meniscectomy, which consists in the partial or total removal of the tissue. And the problem with this approach is that over 50% of the patients undergoing meniscectomy will develop osteoarthritis in the long term. Now, to date, there are a couple of options for meniscal replacement that are on the shelf. Menaflex is an organic-based surrogate, and another synthetic alternative is Activate. Both alternatives have proven to work fairly well in the short term, but in the mid-long term, these materials degrade and the results become similar to those of meniscectomy. And this is why they are not very popular. This eventually highlights the clear need for a new tissue engineered constructs that can replace the damaged native tissue. Now, in order to develop this construct, one should first understand structure and composition of the meniscus. It is a fibrocartilaginous tissue composed of 70% of water, and the rest is collagen and glycosaminoglycans with a ratio of about 3:1 in terms of collagen to glycosaminoglycan. The collagen is organized in fibers mostly running in the circumferential direction of the tissue. During body activities, the meniscus is exposed to compressive, shear, and tensile loading. In particular, it is believed that high shear stresses are associated to the formation of the tears. Therefore, understanding the biomechanics of the meniscus helps understanding tissue pathophysiology and to determine what structural and mechanical features a surrogate should have to survive in a mechanical environment of the tissue. As a tissue, the meniscus is only partly vascularized. The vascular region is also called the red zone and accounts for 10 to 30% of the outer region of the meniscus. The meniscus self-repair capabilities are limited only to the red zone. This means that if a damage occurs in the white zone, it will not heal by itself. We have previously shown that nutrition of cells in the white zone mainly relies on diffusive transport from the synovial fluid in the knee. And this is why the understanding of biotransport can help us understanding what are the features a surrogate should have in order to guarantee appropriate nutrition to cell and to design drug delivery therapies. Now, let's start first on focusing on the biomechanics of the meniscus. It is probably useful to conceptualize the structure and the composition of the material before we enter into the details of the determination of its mechanical properties. Now, the meniscus has three main components: a viscoelastic network of proteoglycans or glycosaminoglycans, a fluid phase mainly composed of water and ions, and a mesh of collagen fibers. Now, the blending of the proteoglycans and the water generates what we call a poroviscoelastic medium. And the collagen fibers serve as a reinforcement. In the same way you would have rebars into concrete, essentially. Now, we have introduced the terms poroelasticity and viscoelasticity. These are two mechanical behaviors that distinguish from plain elastic behavior that you may be more familiar with. In some cases, poroelasticity and viscoelasticity are confused or mutually exchanged, but they are quite different among each other. In general, both poroelasticity and viscoelasticity cause time-dependent response of the material to a mechanical load. However, the poroelasticity occurs when there is an interaction between the fluid phase, like the water in the meniscus, and a porous solid, like the polymeric network of proteoglycans. Now, this mechanism of interaction is based on friction, which causes dissipation of the mechanical energy. And this basically grants the damping properties of the meniscus in dissipating mechanical load. It's also important to remember that poroelastic phenomena occur on a time scale from tens to thousands of seconds. This is a very important detail at the moment that we want to characterize the mechanical properties of the meniscus in relation to its poroelastic behavior, and we will see it in the next slides. In contrast, viscoelasticity is due to the rearrangement of polymeric chains in the solid matrix during the mechanical loading. And the driving force of this phenomenon is entropy. Importantly, viscoelastic effects occur on a time scale of cents of seconds to a few seconds. Now that we have clarified the difference between poroelastic and viscoelastic behavior, it is easier to discuss the compressive behavior of the tissue. This is generally investigated via tests of stress relaxation. In a stress relaxation, tissue samples are placed into a chamber that is surmounted by a porous plug. The tissue is then compressed, and because of the compression, the fluid into the tissue squeeze out in a similar way as you would think about a sponge that is full of water that is squeezed. Now, by looking at how the stress redistributes into the sample over time, one can determine the compressive elastic modulus of the material, as well as what we call the hydraulic permeability, which indicates how easy the fluid moves into the tissue. So compressive modulus and hydraulic permeability can entirely describe the poroelastic behavior of the system in compression. Now, it is important to remark that these tests are done very slowly. This is done in order to neglect all the potential viscoelastic phenomena that may superimpose. Think about that the stress relaxation is generally performed in a time frame of 1 hour. And the viscoelastic effects, as I mentioned before, occur on a time scale of a few seconds. So by testing slowly, we can purely address the problem of poroelasticity, neglecting the viscoelasticity. Now, we have conducted these experiments both on human and animal tissues. We have done extensive experiments. As you see in this slide, what we generally do, we take the meniscus, we cut it in wedges, and then we start to slice slices of the tissue with the compressor that you may be familiar with. And once we obtain the desired thickness of the slice for this experiment, which is between 1 to 2 millimeters, we use a corneal trephine to punch the slice and obtain a cylindrical specimen that you see in the central figure, the white cylinder. And then we have built our custom-built uniaxial apparatus to conduct mechanical compressions. And through computational model, we extract from the experimental data the values of compressive modulus and hydraulic permeability. Now, in our test, we cut samples along 2 orientations. The axial orientation, in which the collagen fibers are parallel to the bases of the cylinder, so in the axial direction of the tissue, and the circumferential samples, in which the fibers run along the axis of the cylinder. Now, we do this because we are interested in comparing potential anisotropic, or if you want, orthotropic properties of the meniscus in compression. Now, here are some summaries of some of the main results that we found in our experiment. Our results show that neither the compressive modulus or the hydraulic permeability are affected by the orientation of the samples. Axial and circumferential sample properties are not statistically different, and this indicates that the fibers do not contribute to the mechanics of the meniscus in compression. We also observed trends indicating increase in the compressive modulus with the level of compression. And decrease in the hydraulic permeability with the level of compression. This is in some way intuitive, because if we keep on compressing the material, we are closing the pores of the porous structure, and so we are creating more difficulty to the fluid to permeate or move into the tissue. And this is the reason why the hydraulic permeability should decrease. Now, in order to investigate the viscoelastic properties of the tissue, we use shear tests. This is done with a rotational rheometer, which applies torsion to our cylindrical samples. The sample preparation for this type of approaches are very similar to those that we have seen for the compression test. Now, even in this case, we evaluated the behavior of axial and circumferential samples. For the axial samples, the fibers are parallel to the plane of torsion. Therefore, they do not contribute to the performance in shear. However, for the samples in the circumferential directions, fibers should contribute to the mechanical behavior since they elongate during shear, as you can see in the bottom image. It is also important to observe that a pure torsional shear test solely measures the viscoelastic behavior of the material. And this is because a pure shear test does not stretches or compresses the overall material. So by not changing the volume of the material, we are not changing motion of the fluid into the sample. And so we are not observing any fluid-solid interaction, which means that we are not observing any Poroelastic phenomenon. So we have extensively investigated the shear properties in several studies on both animal and human meniscus and the relationship with the tissue and structure. We've also investigated different regions of the tissue. In particular, in this slide, in the bottom figure, you see preparation of samples in 3 main regions. From the central wedge of the meniscus, we extract samples on the top surface. On the bottom that we call the femoral layer, which would be a layer of cartilage that wraps the meniscus and that is in direct contact with the femur. We take samples from the tibial layer, that is the bottom layer of the cartilage that wraps the meniscus, and we also take samples from the core, which is purely fibrocartilaginous, where the collagen fibers are. Now, here are some of the main results that we found. In the top diagram, there is a comparison in terms of shear properties of axial and circumferential samples. And you can see that the complex shear modulus of the circumferential samples is significantly larger than the axial samples. And this confirms our hypothesis that fibers should contribute to the shear properties of the meniscus. On the bottom, there is a comparison of the shear properties of various layers of the tissue for axial samples. It can be seen that the femoral layer has significantly lower shear modulus than the other regions of the tissue. And this is a very important finding because it may explain why meniscal damage frequently originates on the femoral surface and then propagates throughout the tissue. Now, we have also investigated the relation of these properties with tissue composition. And we have seen that for all the regions of the tissue that we investigated, the shear properties in terms of both loss modulus and elastic shear modulus, decrease with the water content. In reality, when the meniscus starts degenerating, its water content increases. Therefore, the reduction of the shear properties with the increase of water may explain the acceleration of the process of tissue degradation that occurs. Because as the tissue degenerates, increases the water content, its shear properties degrade, and the meniscus is more prone to degeneration. So in conclusion, we were able to show some of the most relevant findings on the Poroelastic and Viscoelastic behavior of the meniscus in relation to its unique structure and composition. And specifically, we have shown how compressive tests can be used to characterize poroelasticity, while shear tests can characterize viscoelasticity. We also shown that collagen fibers do not significantly contribute to the compressive properties of the tissue. However, they significantly increase the torsional stiffness of the meniscus. Now, lower shear stiffness of the femoral layers of the meniscus may explain why tissue damage the tears frequently originate on the femoral side of the tissue. And the negative trend of mechanical properties with the tissue water content may explain the decrease of tissue mechanical performance as the meniscus degenerates. And this pretty much concludes my portion on the biomechanics. And now I would like to switch gear on biotransport, which is equally important for developing strategies to repair or healing the tissue. I will mainly discuss diffusive properties and solubility. Now, diffusivity, the study of diffusivity is something that we have undertaken in several fibrocartilaginous tissues. And when measuring the diffusive properties, we use a very unique technique that we partly developed in our lab. And this technique is called fluorescence recovery after photobleaching, or also the acronym is FRAP. The basic principle of FRAP is quite straightforward. We want to study the motility of molecules into a tissue. And in order to do that, we need to use our molecule of interest needs to be fluorescently labeled. Now, we do this. This is a laser scanning microscopic technique. So at a certain point, we can illuminate a spot of the tissue with a very high intensity laser light. This causes the bleaching of the fluorescent molecules into the spot. And after the photobleaching, the molecules will diffuse into the tissue of the bleached spot, and the bleached spot will disappear. Now, by monitoring how fast the bleached spot will disappear, we can determine the mobility of the molecules into the sample, and so the diffusivity. Now, this seems very straightforward, but there are some problems that arise when we deal with a material like the meniscus, because in practice, what we generally do, let's say the theoretical approach to this technique is to transform the diffusion equation into the Fourier space. This provides a solution that indicates that the Fourier transform of the molecular concentration in a region of the sample exponentially decays with the diffusion coefficient. Therefore, we acquire a time series of images after bleaching, monitoring the recovery, and we transform these images in the Fourier space, and then we curve fit the fluorescent intensity decay, which is proportional to the concentration of the molecules, with the transformed image in the Fourier space with our model equation in order to yield the diffusion coefficient. Now, this approach works again if the diffusion is isotropic. Now, in the case in which the diffusion is anisotropic, as we will see in the meniscus, we need to determine, instead of a diffusion coefficient, a diffusion tensity. Now, this means that we have three unknown parameters to find, and this requires three independent measurements. Now, the good news is that when the diffusivity is anisotropic, the bleach spot during recovery forms an ellipsoidal shape. This is due to the fact that diffusivity may be different in different directions, and this is the reason why the spot obtains this oblongated shape. Now, this loss of circular symmetry can be exploited in order to develop independent measurements. In fact, we can take any FRAP image and conduct two different recovery analyses on two adjacent quadrants of the image. And this would give us two independent measurements. The third independent measurement could be the orientation of the spot. So through these three independent experimental measurements, we can determine all the components of the diffusion tensor. Now, we conducted several studies on diffusivity of the meniscus, and we have investigated the diffusive properties of molecules as small as glucose, which is the main nutrient into the tissue, to molecules of the size of anabolic growth factors like IGF-1, or to larger cytokines. We've also investigated the diffusivity at different level of tissue compression because the meniscus is generally subjected to compressive loads. So we were interested into understanding how this can affect the diffusivity. Now, the sample preparation techniques that we use for the samples is similar to those that we have seen in the previous experiments. Again, we take wedges of the meniscus, we slice it with a compressed foam, and then we use corneal threadings in order to cut perfectly shaped cylindrical samples. And in order to conduct experiments on diffusivity in compression, we developed a custom-made compression chamber where we introduce the sample. And this chamber is mounted on a glass slide, so it allows us to investigate the diffusivity as we progressively increase the level of compression of the sample. Now, here is a summary of the most relevant results that we have found. In the top left diagram, you see a comparison of the diffusion coefficients for several molecular probes of different size. And you see that the diffusivity in the direction parallel to the parallel and orthogonal to the fibers is investigated. And it's found that it is faster in the direction of the fibers. So diffusivity seems to be faster in the direction of the fibers versus the direction orthogonal to the fibers. This, it is also observed in that the anisotropic ratio between different diffusivity in parallel and orthogonal direction is not affected by molecular size, as you can see in the diagram on the top right. Now, in the bottom left, instead, we show how diffusivity reduces as the molecular size increases, following a well-known Ogston model for diffusivity. And finally, in the bottom right diagram, we show how diffusivity significantly reduces as tissue compression increases. Now, this is in some way intuitive after we clarified that the diffusivity reduces with the increase in the molecular size, because it's a matter of interaction of the molecule with the structure. If we compress, the pores into the structure become smaller, and so molecules find more difficult to traffic within the tissue. So while most of these results are intuitive, the peculiar question here is why molecules move faster along the fiber direction when compared to the orthogonal direction. And in order to answer this question, we conducted a scanning electron microscope analysis on the surface of the samples that were cut in the direction orthogonal to the fibers for the circumferential sample and parallel to the fibers. So in the top image, you can see a circumferential section, and you can see pores. These pores extend along the direction, the circumferential direction of the tissue, and we call them microtubes. In the past, we have found similar microstructural arrangements in other fibrocartilage, like the annulus fibrosus of the intervertebral disc. Obviously, these pores are not present into the axial sections that would run along the direction of the fiber. So we hypothesized that the microtubes may represent preferential pathways for molecular movement into the tissue. And this explains why diffusivity is faster in the direction parallel to the fibers. We also investigated solubility, since solubility properties are very important. In particularly, we are interested into determining the partition coefficient, which is the proportional concentration of a molecule within the tissue relative to the concentration in a free solution. Now, understanding the solid partitioning in the meniscus tissue is beneficial for developing effective strategies for drug delivery to the tissue for the treatment. The objective of our research was to investigate the role of solid size on the partition coefficient in various regions of the meniscal tissue. And again, we focused our analysis on samples extracted from the core of the meniscus as well as the femoral and tibial layer. In order to calculate the partition coefficient, we immersed the tissues in solutions of fluorescent probes and measured the attenuation of the fluorescence of the solution due to absorption of molecules from the tissue. Then we placed the tissues in another bath of purely PDS and measure the concentration of fluorescent solid discharged by the tissue. Finally, we measure the water volume into the tissue in order to determine the actual volume in which the molecules were solubilized once in the tissue. Based on this data, we used an established equation in order to determine the values, the partition coefficients for molecules as small as the glucose or large as growth factors or larger proteins. Now, not surprisingly, our results indicate that solubility decreases as the molecular size increases. However, we did not see any significant difference in solubility across tissue layers. This is important and means that the external meniscal layers, like the femoral and tibial layers, do not contribute as a potential barrier to the solubility of drugs delivered from the synovial fluid. And interestingly, we also found that the partition coefficient decreases with the water content, which also makes sense because decreased water content means less volume available for molecules to be solubilized. So in conclusion, we found that diffusivity in circumferential direction of the meniscus is about twice as faster as that of in the axial or radial direction of the tissue. Diffusivity reduces also as the molecular size increases and or the tissue compression increases. Now, the unique meniscal structure, these microtubes that we found, may be responsible for this behavior. Finally, the relationship among molecular size and transport properties have implication on kinetics of drug delivery and effectiveness of therapeutic approaches. In practice, smaller molecules penetrate more and spread faster, and this may increase their therapeutic effect. So these findings definitely provide some important guidelines in our opinion on possible carriers for drugs into the tissue. And so this is all, and I would like to thank you for your attention. And as concluding remarks, I would like to acknowledge all the students that were involved into the studies, as well faculty, and obviously NIH being the sponsor of this research. And thank you.
Oh my gosh, thank you so much, Francesco, for your talk on the meniscus and your studies on it. And I want to open the floor now to see if anybody has any questions. My colleague Chloe and I have been reading some of your academic papers, and we certainly have a few questions we would love to ask, but I want to open up the floor to see if anybody else does first. And feel free to unmute yourself and show your video to ask as well. If not, maybe Chloe, do you want to jump in with a couple of our questions?
Hello, can you hear me?
Yes, we can.
Okay,
go for
wonderful. So the first question that I have, first I want to say thank you so much for the interesting webinar. But my first question was, I was wondering, how does the water content and composition of the collagen in the meniscus differ in someone who is young or athletic versus someone who is older?
There are several data in the literature in general. We have seen that with the level of degeneration and with the age, there is going to be an increase in the water content into the tissue, which is actually the opposite of what generally happens into cartilage or intervertebral disc. And there is generally degradation of the content of glycosaminoglycans that are present. So you really see the difference. There is a major difference also in the aspect of the meniscus that after degeneration, of course, there is an initial phase of degeneration where the water content increases, but then when the meniscus starts to completely degenerate and the proteoglycans leak out, then there is a shrinkage of this tissue. So if you compare a young athlete, well, if you compare a young person, you will see that the meniscus is plump and whitish. And if you see an elderly individual, you will see a very thin layer that is yellowish. Now, young athletes undergo significant mechanical loads that this can accelerate the process of degradation of the tissue as well.
Thank you.
Wonderful. Any other questions?
So, hello, thank you very much. I don't know much about meniscus, and also the presentation you gave helped me. I was receiving email from Precisionary instrument, and then I saw this opportunity for learning about this meniscus research. I was interested in this one because I myself got meniscus injured about 2 months ago. So I still have that symptom going. By the way, I'm a reproductive biologist, a professor of University of Illinois. So I have nothing to do with this area, but because of the injury, I was interested in. One obvious question from me as a patient, and I'm 60 years old, and my injury is in the red zone, I think. Whether that can be healed, that's my primary question, by without having surgery, by having invasive treatment, any invasive treatment. For example, I heard about PRP injections and hyaluronic acid injection to reduce the friction in the area. So I guess this is not your area, but if you have any thought, that will be helpful.
Okay, it's not exactly my area, but we also work on strategies for conservative approaches that would try to limit the use of surgery. Obviously, the use of surgery, if it's in the red zone, there is high chance of self-healing. In general, we saw a recent paper published this year that shows that the highest results in terms, the best outcomes for patients in the treatment of the meniscus is not to go through surgery immediately, but to start a conservative approach of physical therapy. If with physical therapy and pain management the problem can be solved, there is probably no need for surgery. And if there is a need for surgery, the outcome of the patients who performed physical therapy before surgery is superior to that to patients that never performed physical therapy and they need to recover. This physical therapy helps strengthening all the muscles around the knee capsule, so help for a speedier recovery after surgery. PRP is generally used in several surgical approaches for knee, because it's known that it has this anti-inflammatory effect and also stimulates the anabolic activity and tissue regeneration. But I am not sure if only PRP and hyaluronic acid injections could actually solve the problem. I guess that this is something that may vary from individual to individual, but certainly the use of physical therapy prior to surgery would, it seems to be the new recommendation from surgeons.
So so the meniscal care, I looked at the MRI imaging myself. I'm not a doctor, but I can see that there is a kind of a crack from the outside toward the inside, and it's not that bad. I walk and I can still run quite well. I went to physical therapy, but the therapist say I may not need therapy. I still need to see the surgeon and to listen what he may recommend. So my question is, when we have a crack like this,
yeah,
can that be healed? That's my question.
I guess that I don't think that I am qualified to give you an educated answer on that.
I understand that, but you as a researcher may be able to say whether the tissue, once the tissue is damaged and crack happen, whether that cannot be healed or can be healed.
In general, with the surgical approach, there are encouraging outcomes on the red zone. So a surgical approach would be probably the one that may guarantee the highest rate of success. But again, I would see a doctor.
I understand. Yes.
And good luck.
Thank you very much.
Good luck, Dr. Ko. Sorry we weren't able to help so much clinically, but the research is very fascinating.
You know, I went to the doctor. I met him 3 times already, and he said he has done about over 1,000 surgeries, and they don't collect the data. And I was asking, so after you do surgery or after you do any other treatment, do you follow them up by taking additional MRI after many months after and so on? They don't do that. They just do the surgery, and then people say that they have improvement in the symptom, then they're happy. But there's no follow-up examination of the tissue itself.
Well,
So,
yeah, this is a matter of, I guess, probably we are entering now into an area that is probably definitely not our expertise, but this is a matter of how insurance would cover the expenses for a further MRI and what would be the benefit of having a further MRI if the patient outcome is positive. So if you want to find the results, if you want to find more information about the outcomes, there are dedicated studies where they have done a specific follow-up on a reduced sample. Obviously, we are talking about generally convenient samples and not studies of like 10, 20 participants, but it's more difficult to find studies on a larger population. This is because generally there is no medical image needed if the patient is reporting improvement. And this is the reason why we don't have MRI samples.
Got it. Well, thank you so much to everybody. If there are no more questions, I think what we'll do is we'll wrap it up. So again, Dr. Travascio, thank you so much for your time on sharing with us your research. We're looking forward to your additional publications, and thank you for also using the compressome for making your tissue sections. And with that, I hope everyone has a wonderful week.
All right, thank you very much.
Bye-bye.
Bye-bye.